Let's talk about cells. Cell theory has three parts: that cells make up all living structures, that cells perform the chemical reactions and functions that maintain life, and that cells come from pre-existing cells. The cells vary in size and shape. In our bodies, there's over 290 different types of human cells. The biochemical functions of cells are dictated by the shape of cells and the specific subcellular structures that are present. Cells can develop specialized characteristics, and when they do, they are said to be differentiated. Continuity of life has a cellular basis. All cells come from pre-existing living cells. Cells are small, they're measured in micrometers. Cells vary in size and shape and function. Here's a small red blood cell and a slightly bigger white blood cell. Here's a fairly typical cell, and here's an elongated muscle type of cell. Neurons have these weird extensions that come off of them. Most of our cells bind to each other to form sheets or rows. The binding of cells can reinforce their function. These cardiac muscle cells bind together in order to squeeze the blood to create pressure to push the blood through the body. Extracellular materials are substances found outside of cells. And there's different classes. There's extracellular fluids, often called body fluids, and they include the interstitial fluids, which we've mentioned before. And those are the fluids that bathe the typical type of cell. But there's other types of extracellular fluid, like blood plasma, that's the fluid in blood, and cerebrospinal fluid, that's the fluid that surrounds the brain and spinal cord. And then there's synovial fluid that's found in joint spaces. Extracellular materials include cellular secretions like saliva and mucus, tears. An extracellular matrix is the substance that acts as glue to hold cells together. We'll talk way more about matrix in Chapter 5 with tissues. There's no typical cell, but a composite cell shows the components found in most cells. Most cells have three major parts: a nucleus that contains genetic material, it directs the cell's activities; cytoplasm that consists of organelles with specific functions, suspended in a liquid called cytosol, which we also call cytosol intracellular fluid; cell membrane, which is the flexible outer boundary. It consists of phospholipids in a bilayer interspersed with proteins, cholesterol, and carbohydrate molecules. So here's a typical cell. It has a cell membrane that is the boundary. And this cell membrane is made up of a phospholipid bilayer. Everything inside the plasma membrane, all the way to the nucleus, is the cytoplasm. Cytoplasm includes cytosol, which is the fluid inside of cells, also known as intracellular fluid. And then all these substructures. These include organelles and inclusions. Notice that endoplasmic reticulum hugs up against the nucleus. There's rough endoplasmic reticulum and smooth endoplasmic reticulum. They send vesicles containing substances to the Golgi apparatus, mitochondria, which produce ATP when oxygen is present. There are lysosomes that digest things, and peroxisomes. There are cellular extensions. There can be short cellular extensions called microvilli that aid absorption by increasing surface area. There's long cellular extensions called cilia, which move substances that are on the surface of the cell, like mucus. Sperm cells in humans have a long extension known as a flagellum that allows the cell to swim. The cell membrane is also known as the plasma membrane, and it's made up of a phospholipid bilayer with cholesterol molecules and proteins. Some of the proteins span the phospholipid bilayer from one side to the next. They're known as transmembrane proteins. You can also call them integral because they're embedded within the plasma membrane. And then we have peripheral proteins that are embedded in the membrane, but they are only facing one side of the membrane. On the external surface of all of our cells, there are carbohydrate molecules bound to proteins. These are known as glycoproteins, and bound to lipids known as glycolipids. These surface sugars form the glycocalyx of the cell. Inside the cell, the cytoskeleton anchors to the plasma membrane, providing stability and structure to the cell. Cell membrane functions include being a physical barrier. The phospholipid bilayer forms the outer boundary of the cell, and it plays a role in cellular activity, which is constantly changing based on cellular needs. It controls what enters and what leaves the cell. The phospholipid bilayer separates the intracellular fluid from the extracellular fluid, and it allows separate activities to occur inside and outside of the cell. The cell membrane functions to be selectively permeable. It regulates entry and exit of substances and waste, but really particularly hydrophilic substances. This means it controls the rate at which certain molecules can enter and exit the cell. The plasma membrane functions in communication with receptors that allow it to carry out certain tasks when certain molecules bind to those receptors. This is known as signal transduction. It permits cells to receive and respond to messages like hormones and neurotransmitters. To transduce means that you take something that's in one form and relate it into another form. For example, if you were to read something out loud, you are transducing the words that were written into spoken words. It would be the same message, but you experience it in a different form. The other plasma membrane function is cell recognition. Various proteins and carbohydrates in the membrane mark the cell so that other cells can identify it. A white blood cell can go into a tissue like the liver and recognize that these cells belong in the liver, that they're performing the functions that are appropriate due to the surface markers found on the cells. This helps us in identifying foreign cells or invading cells, like from cancer. Cell membrane components include the phospholipids that make up the phospholipid bilayer. They have water-soluble or hydrophilic heads that form the surfaces inside and out, and the water-insoluble hydrophobic tails that form the interior. The bilayer is permeable to lipid-soluble substances, but not permeable to water-soluble substances. Other cell membrane components include cholesterol, which stabilizes the membrane and helps keep it impermeable to water-soluble substances. Some lipids and proteins can move, and this gives our cell membrane fluid mosaic properties. Membrane proteins have many functions. There's pores, channels, receptors, enzymes, cell contact and identification proteins, and cell adhesion molecules, also known as CAMs. Protein receptors respond to extracellular signals, like hormones and neurotransmitters. Pores, channels, and carriers transport small molecules and ions, and they can transduce signals. Enzymes catalyze chemical reactions. Cell surface proteins identify and establish self. Cellular adhesion molecules enable cells to stick together. Mutations in sodium channels can cause inability to feel pain or extreme pain conditions. Mutations in potassium channels can disrupt electrical activity of the heart and disturb heart rhythm and or impair hearing. Abnormal chloride channels are the cause of cystic fibrosis, which has many signs and symptoms, including production of thick mucus, which causes difficulty breathing, a clogged pancreas, and salty sweat. These diseases illustrate the importance of these transport proteins embedded in our cell membrane. Cellular adhesion molecules link cells together to form tissues. They can also guide cells that are on the move. So let's look at some examples of that. Selectins coat white blood cells and anchor them to capillary walls by providing friction. Adhesion receptor proteins bind to integrins to help white blood cells leave a capillary. Integrins direct white blood cells through capillary walls towards infection sites, and other cellular adhesion molecules guide embryonic cells towards maternal cells to form the placenta, and they can establish connections between nerve cells. This is an image of an endothelial cell. This is the type of cell that lines all the vessels of our body. That includes blood vessels and lymph vessels. This image shows the carbohydrates that are found on the surface of the cell membrane, and it gives it a hairy appearance. Every cell type has different patterns of this carbohydrate sugar coating. It functions as specific biological markers for cell-to-cell recognition, allows the immune system to recognize self versus non-self, and allows the body to distinguish between its own healthy cells and transplanted tissues, diseased cells, or invading organisms. Imbalance of homeostasis occurs in cancer cells, in which the cancer cells can change the glycocalyx so rapidly that the immune system cannot recognize that the cell has been damaged and needs to be destroyed. As a result, the mutated cells are not destroyed and they are able to replicate. Cytoplasm is located between the plasma membrane and nucleus. It is composed of cytosol, also known as intracellular fluid. It's water with thousands of solutes. It includes proteins, salts, sugars, amino acids. Organelles are metabolic machinery of the cell, each with specialized function. They can be membranous or non-membranous. The membranes allow crucial compartmentalization. The membranous organelles include mitochondria, endoplasmic reticulum, Golgi apparatus, lysosomes, and peroxisomes. Inclusions are grouped or storage of chemical substances in the cytoplasm. These are things that are grouped together, but they're not like set apart, like in a closet. They're just kind of together, but off in a corner. They vary with cell type, but they include glycogen granules, pigments, lipid droplets, vacuoles, and crystals. The cytoplasm also contains the cytoskeleton, a supporting framework of protein rods and tubules like scaffolding that gives the cell shape. This is the model that we have in the lab. I labeled the parts for you, and it's useful to refer to as you study. Within the cytoplasm, we find ribosomes. Ribosomes are composed of protein and ribosomal RNA. They can be found roaming free willy-nilly around the cytoplasm or embedded like studs on the rough endoplasmic reticulum. On the ER, they synthesize proteins to be incorporated into membranes, lysosomes, or exported from the cell. The function of ribosomes is protein synthesis. Only ribosomes can synthesize proteins. And wherever ribosomes are found, we give that area credit for protein synthesis. How they work is they provide structural support and enzyme activity to link amino acids together by peptide bonds in protein synthesis. The endoplasmic reticulum hugs the nucleus all around the cell. And the endoplasmic reticulum is made up of these flattened sacs that look something like pita pockets to me. The rough endoplasmic reticulum appears rough because of the embedded ribosomes. The smooth endoplasmic reticulum lacks ribosomes. The smooth and rough endoplasmic reticulum are connected. Endoplasmic reticulum are membrane-bound sacs, canals, and vesicles. They're interconnected tubes and parallel membranes that enclose fluid-filled spaces known as cisterns. They are continuous with the outer nuclear membrane. There's a tubular transport system that links them together, and there's two varieties, rough and smooth. Rough endoplasmic reticulum has ribosomes embedded on its membranes, so it conducts protein synthesis. It produces most cell proteins. It manufactures all secreted proteins. It also synthesizes membrane integral proteins and phospholipids. The simple proteins move from the endoplasmic reticulum interior to be enclosed in a transport vesicle and sent to the Golgi apparatus. Smooth endoplasmic reticulum does not have ribosomes, however, it is full of enzymes. The enzymes of the smooth endoplasmic reticulum are integral, embedded within the walls of the membrane. They function in lipid metabolism, cholesterol and steroid-based hormone synthesis, making lipids of lipoproteins, absorption, synthesis, and transport of fats, detoxification of drugs and pesticides and carcinogenic chemicals, converting glycogen to free glucose, and the storage and release of calcium. The Golgi apparatus receives vesicles from the endoplasmic reticulum. Here's some rough endoplasmic reticulum. It synthesizes proteins, then buds them off into a transport vesicle that is carried to the Golgi apparatus. The Golgi apparatus will sort and modify and repackage the materials and send them to where they need to go. Some will need to go to the exterior of the cell, so they will be sent in secretory vesicles to the surface of the cell to be released by a process known as exocytosis. Vesicles are membranous sacs that store or transport substances. The Golgi apparatus is flattened, stacked, membranous sacs. It's a lot like a post office, but it modifies its contents as well, which the post workers shouldn't do. So the Golgi apparatus takes in substances that were sent to it through transport vesicles. It modifies, concentrates, and packages them, mainly its proteins and lipids, from the endoplasmic reticulum. They're tagged for where they need to be delivered, sorted and packaged in vesicles, and the vesicles will bud off from the Golgi. They'll go in secretory vesicles to be released for export by exocytosis. Some of the vesicles of lipids and transmembrane proteins are exocytosed in order to become embedded in the plasma membrane or in the membranes of other organelles. And lysosomes are forms containing digestive enzymes, and they remain inside the cell. For cells that produce milk in the mammary glands, proteins would be made in the rough endoplasmic reticulum, and some lipids would be made in the smooth, and they would send vesicles to the Golgi apparatus so that the Golgi could modify and repackage and concentrate and tag them for delivery. Then they would be delivered to the exterior through secretory vesicles for exocytosis. A mitochondrion is the powerhouse of the cell, or plural, they're known as mitochondria. They are double membrane fluid-filled structures with inner shell-like cristae. These are the folds you see inside. The chemical reactions that extract energy from nutrients like carbohydrates, proteins, fats, and alcohol in the presence of oxygen occur within the mitochondria. This is known as aerobic cellular respiration. It produces ATP and, as a byproduct, toxic waste known as carbon dioxide. Each mitochondria contains its own set of DNA, which makes its own RNA, and they even have ribosomes that are more similar to the ribosomes of bacteria than they are to the ribosomes found elsewhere within our cells. When we need more mitochondria, they just divide in the same process that bacteria divide, known as fission. Lysosomes are small spherical membranous sacs that contain enzymes that digest things like proteins, carbohydrates, nucleic acids, bacteria, debris, worn out cell parts like old mitochondria. They destroy cells in injured or non-useful tissues known as autolysis. They are the garbage disposals of the cells. They can even break down bone to release calcium. They're made up of a membrane, and they contain enzymes known as hydrolases that digest stuff. Peroxisomes are also membranous spherical sacs, similar to lysosomes. However, they contain different types of powerful enzymes, known as oxidases and catalases. That allows them to digest lipids, alcohol, and hydrogen peroxide. They detoxify harmful or toxic substances. They can catalyze and synthesize fatty acids. They neutralize dangerous free radicals. Free radicals are highly reactive chemicals with unpaired electrons that act like flubber. Oxidases convert free radicals to hydrogen peroxide, which itself is toxic, but then the catalases convert the hydrogen peroxide to water and oxygen, which are safe for our cells. Microfilaments, microtubules, and intermediate filaments are structures in the cytoplasm that create the cytoskeleton. They play an important role in maintaining cell shape and structure, promoting cellular movement, and aiding the cell in its functions. This is a cell that had the microfilaments dyed green, and it looks like a spider webbing that takes up most of the space of the cell. And then the intermediate filaments were dyed this red color, and then the microtubules were dyed this brown. These provide the scaffolding, structure, and aid in organization of the cell. Microfilaments are tiny rods of actin protein. They provide cellular movement such as muscle contraction. They also strengthen cell surfaces and help resist compression. Microtubules are larger tubes of tubulin. Their rigidity maintains cell shape and help cells resist pulling forces. They make up cilia, flagella, and centrioles and help to move organelles. Many substances are moved throughout the cell by motor proteins, which use microtubules as tracks. Intermediate filaments are composed of several proteins. They support the nuclear envelope. They determine the overall shape of a cell and the distribution of the organelles. You can see how the protein rods hold things in an organized manner so that they have the correct shape and spacing, and they connect the organelles, which is why they can be used as tracks for the motor proteins. The microtubules of microfilaments allow the motor proteins, like this little guy here, to walk vesicles full of substances to the correct spot. How it works is the organelles, like the Golgi apparatus, hold a vesicle in just the right spot so that this motor protein, when he's walking along the track, will pick it up and then he carries it with him, and this track leads to where this vesicle needs to go. The feet move because an ATP binds to the foot, is broken down to release energy, and that energy allows the foot to be thrown forward. And then another ATP binds to the other foot, which throws it forward. And in this manner, materials are moved through our cells. I think these are super cute. The centrosome is found near the middle of the cell, right near the nucleus. It consists of two centrioles. Centrioles are cylinders composed of microtubules. They will produce spindle fibers for cell division, which allow us to distribute chromosomes properly. The centrioles are the basis for flagella and cilia. Cilia are motile extensions of the cell membrane. They form a fringe on the surface of certain epithelial cells. Cilia are longer than microvilli but shorter than flagella. They move in a pattern that allows them to move a substance that is external to the cell, like mucus, in one direction. The cilia in your nose move the mucus to the back of your throat, and the cilia in your trachea move the mucus up the trachea to the back of your throat. So they can move stuff like mucus in the respiratory tract, and they can propel the egg towards the uterus along the uterine tubes. Flagella are similar in structure to cilia, but they are much longer. They propel the whole cell and allow it to swim like an eel. The tail of a sperm cell is the only flagellum on a human cell, and each cell has only one flagellum. Microvilli are tiny extensions of the cell membrane. They're composed of actin, and their purpose is to increase surface area for absorption. So where do you absorb things? That's mainly going to occur in the small intestine. That's where we absorb the vast majority of the nutrients from our diet. Some diseases that can occur on the organelle level include Malus, which is a mutant gene in DNA of mitochondria. As a result, the person cannot extract the maximum energy from nutrients. Krabbe disease, caused by inability to produce one lysosomal enzyme, so the person cannot produce myelin for nerve cells, which leads to severe damage to the nervous system. Adrenoleukodystrophy, or ADL, is caused by a lack of a protein in the membrane of peroxisomes. Fatty acids build up, destroying myelin sheaths and nerve cells, and the nerve cells cannot transmit nerve impulses fast enough. The cell nucleus is the largest organelle. It's the genetic library with blueprints for nearly all cellular proteins. It responds to signals, dictates kinds and amounts of proteins to be synthesized. Most of our cells are uninucleate, meaning they have one nucleus. But some cells, like skeletal muscle cells, bone destruction cells, and some liver cells are multinucleate. Red blood cells are anucleate, meaning they have no nucleus at all. The nucleus is found in the center of the cell, and it has a double membrane like a mitochondrion does. They both contain DNA. Remember, the mitochondria had its own DNA. There's nuclear pores that allow stuff to enter and leave the nucleus. There's a dark spot within the nucleus known as the nucleolus, and the fluid inside the nucleus is known as nucleoplasm. The DNA inside the nucleus is in the form of chromatin unless cellular division is occurring. The nucleus contains genetic material and controls cell activities. The nuclear envelope is a double-layered membrane surrounding the nucleus. It separates nucleoplasm from cytoplasm, and nuclear pores allow passage of certain substances. The nucleolus is a dense, dark body of RNA and protein. and it is the site of ribosome production. Chromatin is the cell's chromosomes wound around histone proteins. DNA spends most of its time in the chromatin form and only condenses down into the chromosomes that we see during cell division. We're going to spend some time talking about movements of substances into and out of the cell. Remember that our cells are surrounded by interstitial fluid. That's the solution that bathes our tissue cells. It is the main component of extracellular fluid, which also includes other fluids like plasma, cerebrospinal, and synovial fluid. Interstitial fluid is found in the interstitial spaces or tissue spaces. That's the spaces in between cells. And it contains thousands of substances like amino acids, sugars, fatty acids, vitamins, hormones, salts, and waste products. The plasma membrane allows the cell to obtain from the interstitial fluid exactly what it needs, exactly when it is needed, and to keep out what it does not need. Plasma membranes are selectively permeable. Some molecules pass through easily, some do not. Selectivity is due to polarity, molecular size, and the presence of gates. It allows regulation of hydrophilic molecules into and out of the cell. There are two ways substances cross the membrane: active processes and passive processes. Simple diffusion is how molecules move through the membrane in between the phospholipids to enter the cell. We can also have diffusion through the membrane if the substance is able to fit through a protein embedded in the membrane. We call these channel proteins. Proteins that move to permit a substance through the membrane, that's known as facilitated diffusion, and it requires a carrier. In passive processes, substances are moving from where there's more of the substance to where there's less. Active transport moves a substance against its gradient from where there's less of the substance to where there's more. This requires energy in the form of ATP and a carrier protein that we call specifically a pump. Passive mechanisms do not require cellular energy. They include diffusion, osmosis, facilitated diffusion, and filtration. Diffusion is natural movement of molecules from areas of high concentration to areas of low concentration. This is referred to as moving down a concentration gradient. Diffusion types include simple diffusion, carrier and channel-mediated facilitated diffusion, and osmosis. Atoms, molecules, and ions will move naturally from where they're more abundant to where they're less. This is due to the constant motion of atoms. Diffusion through our cell membrane will occur only with substances that the membrane is permeable to, like oxygen, carbon dioxide, and certain lipid-soluble substances. A sugar cube dissolving in water is an example of diffusion. Solvents in water can diffuse across a membrane that is permeable to both of them until they reach equilibrium. When they reach equilibrium, the molecules still move. They just move such that there's still about an equal amount of elbow room around each. So the concentration of water and solute are equal in both compartments. An imbalance of homeostasis can occur if the plasma membrane is severely damaged. Substances diffuse freely into and out of the cell, compromising concentration gradients. This could occur in burn patients. They will lose precious fluids, proteins, and ions that weep from their damaged cells. So diffusion is when we take a substance that's concentrated, like the sugar cube, and put it into a solvent. The molecules all have a random high-speed movement due to their intrinsic kinetic energy. and that movement results in collisions between the molecules. Molecules in higher concentration areas collide more often, resulting in molecules being scattered towards lower concentration areas. The movement of these substances until they're all about evenly dispersed with even elbow room between each, and it occurs until we reach equilibrium. Now, diffusion can occur through just a space, like through this beaker, but it can also occur through a cell membrane. This image shows diffusion through a semipermeable membrane. This membrane allows both the solute, shown in red, and water to pass through. Initially, there's pure water on one side and solute mixed with water on the other side. Because they can both pass through the membrane, they will move until they're evenly distributed across both sides. Molecules have a natural drive to diffuse down concentration gradients, including ones that exist between extracellular and intracellular areas. The speed or rate of diffusion is influenced by concentration gradient, molecule size, with smaller ones moving swifter, temperature, with higher temperatures also increasing the rate of movement, and then properties of the solvent also can affect the speed. A molecule will passively diffuse through the hydrophobic plasma membrane core if it is lipid-soluble or nonpolar like the membrane core, if it's small enough to fit through a membrane channel, or if it's assisted by a carrier molecule embedded in the membrane. Simple diffusion occurs when lipid-soluble solutes move through the phospholipid bilayer into the cell, down their concentration gradient. Oxygen, which is high in blood arriving at a capillary bed, will diffuse from the blood through the interstitial fluid into the cell to be used in the mitochondria to produce carbon dioxide. And carbon dioxide, which is elevated in the cell, will go down its concentration gradient through the interstitial fluid into the blood to be taken back to the lungs to be exhaled. This type of simple diffusion occurs only with substances the cell membrane is permeable to, like water, carbon dioxide, and other lipid-soluble substances. Facilitated diffusion is movement of a substance through the membrane facilitated by the presence of proteins embedded within the cell membrane. Diffusion takes place through these channels and carriers, and the substances are moved from where there's more of the substance to where there's less of the substance. This is used for substances that cannot diffuse across the selectively permeable membrane since they are hydrophilic, or another way to say that is they are not lipid soluble. We use this for water-soluble substances like sodium, potassium, chloride, glucose, amino acids. It is a passive process and no ATP is required. High concentration of this substance allows it to go down its concentration gradient through this carrier, which changes shape as it moves across the membrane and pushes it to the inside of the cell. Carriers are integral proteins that span the cell membrane and change shape as the substance moves across the membrane. A channel is an embedded transmembrane protein that has a pore, P-O-R-E, a pore, a canal, through which the substance passes. Now these carriers and channels are specific in their size and shape. They would let certain molecules or ions through based on their chemical properties like charge, polarity, and size. Carrier-mediated facilitated diffusion uses carriers embedded in the cell membrane. Each carrier transports specific polar molecules. So a carrier that is specific for galactose would not permit fructose through it, even though fructose and galactose look fairly similar. If I wanted fructose to go through the membrane, I would need to make the carrier that's specific for fructose. Binding of a molecule causes the carrier to envelope it and change shape. That results in the molecule being moved across the membrane in that process. Binding of the transported molecule is limited by the number of carriers present. If I have 10 carriers, then those carriers can only move 10 molecules across at a time. We say that carriers are saturated when all are bound to molecules and busy transporting a substance. My cell could make more of that particular type of carrier so that substances could be moved quickly across the cell membrane through them. Channel-mediated facilitated diffusion uses channels with aqueous-filled cores formed by these transmembrane proteins. They move a substance from high concentration to low concentration. They tend to move things like sodium, potassium, and water, though when water moves, we call that osmosis. Their specificity is based on the size of the pore and the charge of it. Water channels are specifically called aquaporins. These channels can come in two types: leakage channels, which are always open, and gated channels, which can be controlled by chemical or electrical signals, but there's times when they're closed and nothing can get through, and times when they are open and substances are permitted to move across down their concentration gradients. Osmosis is movement of a solvent, not solutes. In our body, the solvent is water, and when that solvent moves across a selectively permeable membrane, that is known as osmosis. Water diffuses across plasma membranes through lipid bilayers, which seems surprising because we said that the lipid bilayer keeps hydrophilic substances out. And hydrophilic substances are substances that are polar and like water. Water is water and water is polar, but because water is so small, it's just one oxygen and two hydrogens, these molecules can squeeze in between the phospholipids, so they can pass through the cell membrane. They don't do so very rapidly, but if you give them time, they will pass through the phospholipid membrane in between the phospholipids. There's also specific water channels called aquaporins, and that can allow lots of water to move swiftly across the membrane. The flow occurs when water concentration is different on the two sides of a membrane. Water will move into a region containing higher impermeant solute concentration. That means water will go towards the solutes that are stuck on a certain side of the membrane if there are more of that solute present. If we have a semi-permeable membrane that allows water, the blue, to pass, but the solute, the purple, not to pass, then water will move through the membrane until there's an even distribution of solute ratio to water. This process of the water moving towards the solutes that are limited in their movement, that is osmosis. Osmosis will continue until the extra height of the water creates enough pressure, known as hydrostatic pressure, that the osmotic pressure that drives the water across the membrane isn't enough to push through due to the extra weight of the water above it. So hydrostatic pressure can cancel out osmotic pressure and prevent osmosis. Equilibrium occurs when there's the same concentration of solute and water molecules on both sides of the membrane. Osmotic pressure is the ability of osmosis to generate enough pressure to lift a volume of water. Osmotic pressure increases as the concentration of impermeant solutes increases in a solution. Water moves toward solutions with higher osmotic pressure. Another way to say that is water moves towards solutes. Tonicity is the ability of a solution outside of the cell to alter water volume inside of the cell. Iso means same, so an isotonic solution has the same osmotic pressure. Cells in an isotonic solution have no net gain or loss of water. There's the same number of solutes outside the cell as inside the cell. So the cell stays the same volume. For human cells, a 0.9% saline solution is isotonic. A hypertonic solution has a higher osmotic pressure. Cells in a hypertonic solution lose water because there's more solute outside the cell. So the water leaves the cell, causing the cell to shrink. The shrinking is referred to as crenation. This is what occurs when you're dehydrated. A hypotonic solution has less solute in the fluid surrounding the cell than there are inside the cell. Water is attracted into the cell, causing the cell to swell, and it can even burst, which is referred to as lysing. I remember a hypotonic solution causes a cell to swell up like a hippo. If we put a blood cell in a 0.9% saline solution, then there would be an equivalent amount of solute outside the cell as there is inside the cell, so the solution would be isotonic, and the cell would maintain its volume. If we were to place a red blood cell in a hypertonic solution, then the water in the cell exits to go towards the higher level of solute concentration outside the cell. That causes the cell to shrink. And if we place a red blood cell in a hypotonic solution, then the water enters the cell where there's more solutes in the intracellular fluid. This causes the cell to swell up and possibly burst. These changes in cell volume can disrupt cell function. Filtration is also a passive process. Filtration is a process that moves water and tiny solute molecules through membranes by exerting hydrostatic pressure. In blood vessels, blood pressure is a type of hydrostatic pressure, and it is used to separate solids from water and other small particles. When blood plasma leaves capillaries, water and small solutes are filtered into the interstitial fluid, but large plasma proteins and blood cells are not. This is how we exchange substances between blood and interstitial fluid. Again, this is a passive process, so no ATP is required. Active mechanisms include active transport, which utilizes pumps, and vesicular transport that uses processes like endocytosis, exocytosis, and transcytosis. Both require ATP to move solutes across a plasma membrane for any of these reasons. The solute is too large for channels, or is not lipid soluble, or is not able to move down concentration gradient. Active transport requires carrier proteins that we call solute pumps. They bind specific substances reversibly, allowing the substance to be moved across the membrane. Some carriers can transport more than one substance at a time. Antiporters transport one substance into the cell while transporting a different substance out of the cell. Symporters transport two different substances in the same direction. Active transport moves solutes against their concentration gradient from low concentration to high concentration, and this requires energy in the form of ATP. There are two types of active transport: primary active transport and secondary active transport. In primary active transport, energy from hydrolysis of ATP causes change in the shape of a transport protein. The shape change causes solutes bound to the protein to be pumped across the membrane. Examples of these types of pumps include calcium pumps, hydrogen, or also known as proton pumps, and the sodium-potassium pump. Secondary active transport requires energy that is obtained indirectly due to ionic gradients created by primary active transport. This illustration is showing a sodium-potassium pump. There is more sodium outside the cell and less potassium outside the cell. Inside the cell there's more potassium and less sodium. The sodium-potassium pump is going to move 3 sodiums against their gradient from the inside of the cell to the outside of the cell. And then it will move 2 potassiums against their gradient from the outside of the cell into the cell. Sodium-potassium pumps continuously pump, moving sodiums outside of the cell where there's already abundant sodium, and potassiums into the cell where there's already abundant potassium. The sodium-potassium pump is the most studied pump. It is involved in primary and secondary active transport of nutrients, the ions. It's basically an enzyme. It's also known as the sodium-potassium ATPase. It pumps 3 sodiums out of the cell and 2 potassiums back into the cell, and uses 1 ATP each time it does so. It allows all cells to maintain fluid volume. It's located in the plasma membrane of all cells, and it's especially active in excitable cells, which are nerves and muscle cells. It maintains the electrochemical gradients, which involve both concentration and chemical charge of ions. Now, leakage channels are also located in the membranes, and they allow sodium to leak back into the cell and potassium to leak out. When they're leaking, they're going down their concentration gradients where they naturally would want to go. And when potassium leaks out, the sodium-potassium pump just pumps it back in. And when sodium leaks in, the sodium-potassium pump just pumps it back out. Because of the sodium-potassium pump constantly pumping and moving 3 positives out and bringing 2 positives, form of potassiums, in, it creates a deficit of charge, which causes the inside of the cell to have a negative charge. Just as if you went to the bank and removed $300 and only deposited $200 into it, eventually you're going to overdraw your account if you continue to withdraw and add money in the pattern of 300 out and 200 in. We utilize these differences in charge inside and out of the cell and the differences in sodium and potassium to cause our nerves to fire, our muscles to contract. It allows us to absorb food from our diet and removes waste from our plasma in the kidneys. Secondary active transport depends on ion gradients created by primary active transport. Energy is stored in gradients and used indirectly to drive transport of other solutes. Because the sodium-potassium pump has so many sodiums forced out of the cell, then sodiums want to go to the inside of the cell where there's less sodium, and the inside of the cell has a negative charge and sodium has a positive charge. Well, opposite charges attract. So sodium wants to go into the cell, down its concentration gradient and towards the opposite charge. Sodium wants to come in really bad. So our body creates transporters that will allow sodium to pull in a substance that is going to go against its gradient, like glucose. Generally, there's more glucose inside my cells than out. So glucose is not going to move down a concentration gradient. Moving into the cell, it's going to move against a concentration gradient. So glucose has no drive to want to enter my cell. However, we can use sodium to drag another molecule, like a glucose. This sodium-glucose symporter will allow sodium to drag this glucose against its concentration gradient into the cell. This process only works because we have invested the energy to create such a sharp gradient for sodium by pushing so many sodiums out of the cell using the sodium-potassium pump. We use this secondary active transport to move some sugars, amino acids, and ions across the cell membrane. It's easy to remember secondary transport if you remember that sodium can come to your party if it brings your hot friend glucose. My cell doesn't actually want sodium. The cell will just pump it out. My cell really wants the glucose, and it's just using sodium to bring it to the party inside my cell. Vesicular transport involves transport of large particles, macromolecules, and fluids across the membrane in membranous sacs called vesicles. It requires large amounts of cellular energy in the form of ATP. Vesicular transport includes endocytosis, which transports substances into the cell inside a vesicle. There's 3 different types of endocytosis: phagocytosis, pinocytosis, and receptor-mediated endocytosis. Exocytosis is transport of a substance out of the cell. Transcytosis is transport into, across, and then out of the cell. And vesicular trafficking is transport of a vesicle from one area or organelle in a cell to another. Phagocytosis is a type of endocytosis that is referred to as cell eating. Phago means to eat. Membrane projections called pseudopods form and flow around solid particles that are being engulfed off and pulled into the cell. We call the vesicle that is formed a phagosome. Phagocytosis is used by macrophages and other white blood cells, and these phagocytic cells can move by amoeboid motion, where the cytoplasm flows into temporary extensions that allow the cell to creep. Pinocytosis is a type of endocytosis that is referred to as cell drinking or fluid phase endocytosis. The plasma membrane enfolds, bringing extracellular fluid and dissolved solutes into the cell. Our cells do this a few times every day to just sample what is outside the cell in the interstitial fluid. Maybe something new is there and we need to add some type of carrier or channel in order to bring it across. It is the main way in which nutrient absorption occurs in the small intestine. Membrane components are recycled back to the membrane in this process. Receptor-mediated endocytosis involves endocytosis and transcytosis of specific molecules. Many cells have receptors embedded in pits, which will be internalized along with the specific molecule that's bound to those receptors. This includes movement of enzymes, low-density lipoproteins, also known as LDLs, iron, insulin, and unfortunately, viruses, diphtheria, and cholera toxins can be taken into the cell using receptor-mediated endocytosis. The benefit of this process is that our cell knows that a specific molecule is present and exactly how much there is due to how many is bound to the receptors. So we can bring it into the cell in the right amount. Once it's in the cell, we will release the substance bound to the receptors and send the receptors back to re-embed them in the cell membrane. Exocytosis is a process where material is ejected from the cell. It's usually activated by cell surface signals or changes in membrane voltage. The substance being ejected is enclosed in a secretory vesicle. The docking process triggers exocytosis. Some substances that are exocytosed include hormones, neurotransmitters, mucus, and cellular waste. With exocytosis, the vesicle merges with the cell membrane and expels the contents to the outside of the cell. Transcytosis involves receptor-mediated endocytosis, followed by exocytosis. It quickly transports a substance from one end of the cell to the other, and it moves substances across barriers formed by tightly connected cells. This is how HIV can move across the lining of mucous membranes like the anus or vagina. So the virus is engulfed using receptor-mediated endocytosis into the cell. It's contained within a vesicle and moved through the cell and then released on the other side of the cell. Now it's crossed our barrier and it's inside our internal space, which should be a sterile environment, but now it has invaders. Here's a summary of the different types of transport that we've discussed. The cell cycle is a series of changes the cell undergoes from the time it forms until the time it divides. The stages of the cell cycle include interphase. During interphase, growth of the cell and maintenance of normal functions occur. Cell division is also known as the mitotic phase. The cell divides in two, and it includes mitosis, which is division of the nucleus, and cytokinesis, which is division of the cytoplasm. We see that the cell cycle is mostly made up of interphase. Interphase starts here. There's a G1, S, and G2 phase of interphase. And then we go into the mitotic phase, which is made up of mitosis, which includes prophase, metaphase, anaphase, and telophase, and cytokinesis, which overlaps with the end of anaphase and telophase. Interphase is a very active period of the cell. The cell grows and maintains its normal functions. The nuclear material, which is the DNA, is uncondensed in a chromatin state. Remember, chromatin is DNA wrapped around histone proteins. The cell replicates genetic material, that's its DNA, in order to prepare for mitosis, which is nuclear division. The cell synthesizes organelles, membranes, and biochemicals to prepare for cytokinesis, which is division of cytoplasm. The subphases of interphase are the S phase, known as synthesis phase. That's where the DNA is replicated. There's G1 and G2 phases. They can be known as growth or gap phases. That's when structures and other molecules are duplicated. The cells that permanently cease dividing are said to be in a G0 phase. And cells that do that are neurons, skeletal muscle, and cardiac muscle cells once they are mature. During the S phase, when we duplicate our DNA, we take the strands of DNA that we have. We have 46 strands of DNA, 23 from your mom and 23 from your dad. They come in pairs, so we have two sets of 23 chromosomes. So you have a chromosome 1 from your mom and a chromosome 1 from your dad, and you have a chromosome 2 from your mom and a chromosome 2 from your dad, and so on. These chromosomes are linear strands of DNA, and during the S phase, we take one strand, like one side of this red chromosome. During the synthesis, DNA replication occurs, and we take this red strand here and make an identical copy of it. As we make that copy, the linear strand forms an X. because we take the copied strand and keep it connected to the strand that we copied it from. We connect them at an area known as the centromere, and they are connected by the connected core. So we have copied this DNA, so there is enough DNA to be 2 chromosomes, but we only call it 1. As long as they're connected, we say that it's 1 chromosome with 2 copies, and we call the copy on one side a sister chromatid, and the copy on the other side a sister chromatid. This gives chromosomes their distinctive X shape. There's different types of cell division, meiosis and mitosis. I'm briefly mentioning meiosis, and we will cover it much more when we get to 102. Meiosis is cell division to produce reproductive or sex cells. They're also known as gametes, but those are the ova, which is the egg, and sperm. The type of cell division we're learning here is mitotic cell division. It produces clones, which means that it produces cells that are just like the cell that they divided from. We call these somatic cells. Somatic cells are any of the cells that make up a living organism other than the reproductive cells. And this is essential for body growth and tissue repair. Control of cell division is crucial, so cells divide when necessary, but do not divide unnecessarily. The phases of mitosis are prophase, where chromatin condenses to form chromosomes. Centrioles form mitotic spindle, and the centrioles move to the opposite sides of the cytoplasm. The nuclear envelope breaks down so that the chromosomes are just exposed in the cytoplasm, and the nucleolus disperses. During metaphase, mitotic spindles from the centrioles attach to the chromosomes and align them along the equator between the centrioles. So in metaphase, the chromosomes are lined up in the middle, M for middle. During anaphase, the chromosomes are separated, and so the chromosome, which had 2 sister chromatids connected at the centromere, during anaphase, the centromere connected core is broken. That separates the sister chromatids, and as soon as they're separated, we call each of them its own chromosome. So one chromosome moves to one end of the cell, and the other one moves to the other end of the cell. And so you can remember anaphase A pulls chromosomes apart, or pulls sister chromatids apart into their own individual chromosomes. During telophase, you can think of T for terminal. It's the end, the last phase of nuclear division. The chromosomes return to their chromatin structure. They uncondense. The nuclear envelope reforms around them, and the nucleoli become visible. Here's a summary of the major events of mitosis. During interphase, the cell grows and does normal maintenance. It prepares for a new cellular division by doubling its DNA so that the chromosomes have an extra set of DNA attached to them, called each side called a sister chromatid. Then whenever it's time for the cell to divide, the nuclear envelope breaks down, the DNA condenses, the mitotic spindle forms from the centrioles that have moved to each end of the cell. The mitotic spindle attaches to the centromere of each of the chromosomes. Once the chromosomes are aligned in the middle of the cell, or in metaphase, when the mitotic spindle breaks the connected core and pulls the chromosomes apart, or in anaphase. During telophase, new nuclear envelopes form around the DNA, which uncondenses back into chromatin. Division of the cytoplasm occurs, dividing into 2 separate cells. Those cells will then go into interphase. Cytokinesis is cytoplasmic division. It begins during anaphase and continues through telophase. A contractile ring of actin filaments pinches the cytoplasm in half and creates a cleavage furrow. Newly formed cells will have identical DNA, and they can have slightly different size and number of organelles. The frequency of cell division is strictly regulated, and it varies by cell type. Skin cells, intestinal cells, and blood-forming cells divide often. Neurons divide a specific number of times and then stop dividing. Chromosome tips are known as telomeres, and they shorten with each mitosis, and that provides a mitotic clock. Fluctuating levels of certain proteins in the cell control the cell cycle, and cells divide to provide more favorable surface area to volume relationships. Hormones and growth factors are external controls for cell division. Contact inhibition causes healthy cells to stop dividing when they become crowded, but tumors can result from a loss of control over the frequency of mitosis. There are two types of tumors: benign, which remain in a local area, and they can enlarge. And then there's malignant. They're invasive, cancerous, they can spread or metastasize. Mutation in two major types of genes cause cancer. Oncogenes are abnormal forms of genes that control cell cycle, but they are overexpressed. And tumor suppressor genes normally limit mitosis, but if inactivated or removed, then they cannot regulate mitosis. This image is showing cancer cells which don't have the normal structure of the healthy cells with cilia. Development in cancer can cause changes in cell size, nucleus size, and organization of a tissue. Cancer cells are able to escape regulatory signals. This can lead to uncontrolled growth, invasion into adjacent tissues, colonization of other organs, and the health suffers when tumors rob blood supply from the normal organs. Characteristics of cancer cells include loss of control of the cell cycle, heritability. A cancer cell divides to form more cancer cells. Transplantability. A cancer cell implanted into another individual will cause cancer to develop. De-differentiation, which is a loss of specialized characteristics. A loss of contact inhibition. Ability to induce local blood vessel extension. This is known as angiogenesis. Angio refers to blood vessels. Genesis is to make. So it induces producing more blood vessels to feed the tumor. Invasiveness and ability to metastasize or spread to other areas. So when enough mutations have affected the cell that it can divide uncontrolled, then it will divide and induce angiogenesis to feed it, robbing other tissues of vital nutrients and energy. Differentiation is the process of specialization of cells. Stem cells are unspecialized. They can divide to form two new stem cells. This is known as self-renewal. They can also divide to form a stem cell, and then the other cell is a progenitor cell. And stem cells can differentiate as many cell types. Progenitor cells are partially specialized stem cells. They're daughters of stem cells. They can divide to become any type of a restricted number of cells. They're known as committed cells. Totipotent are daughter cells that can specialize to become any cell type or the cells produced in very early embryo development. Pluripotent are daughter cells that can become a limited number of cell types. These include stem cells of later development and progenitor cells. We have stem cells in our stomach that can make a number of cells within the lining of the stomach, but they can't make completely other types of cells like blood cells or skin cells, just things related to the stomach. So this is showing how an egg and sperm create a fertilized egg, which will undergo cell divisions. And these early cell divisions form stem cells. The stem cells divide to make more stem cells and some progenitor cells. The progenitor cells go on to become committed to being certain cell types. So they differentiate into specific cell types over time. The progenitor cells form even more specific cell over time until we end up with 290 types of cells in our human bodies. Stem and progenitor cells are required for growth and healing. We study them so that we can learn more about the body and aid us in treating disease. There's regenerative medicine that uses the body's ability to regenerate by forming new cells to treat diseases and injury. We can find stem cells for research by using donor stem cells from umbilical cords and stem cells from a patient, either from natural sites like in the bone marrow or reprogrammed differentiated cells grown in cell culture. Apoptosis is programmed cell death. It is a normal part of development. For example, our skin cells are designed to die and be shed when they reach the surface of our skin. Apoptosis is a continuous, controlled, stepwise process. The process begins with mitochondrial membranes leaking chemicals that activate enzymes called cataspases. The cell degrades and shrinks, and then it's phagocytized by macrophages. Macrophages are Pac-Man-like cells that wander our body, eating up things that don't belong and debris. We use apoptosis to remove webbing between fetal fingers and toes. It's protective, like to peel away the skin cells after a sunburn. Necrosis is cell death from damage, and it is not a normal process. It's usually due to disease, injury, or failure of the blood supply. Hyperplasia is the enlargement of an organ or tissue. It is caused by an increase in the reproductive rate of cells growth, or often in the initial stage of the development of cancer. Hypertrophy is enlargement of an organ or tissue from an increase in the size of the cells, and it can include extracellular material around the cells. Hypertrophy means form. In hypertrophy, we have the same number of cells. The cells are just bigger, causing the tissue and organs to be bigger overall. Whereas in hyperplasia, we had an increase in cell number. Atrophy is a decrease in size or wasting away of a body part or tissue. So cell death occurs whenever the mitochondria release chemicals that trigger the process of autolysis, and eventually the parts are consumed by macrophages to remove the debris.